Hello friends, today we will be talking about the preservation potential of the organisms. That means the way in which the organism or any form is preserved as fossil in the rocks. As we all know that the fossils are found mainly into the sedimentary rocks. The depositing layers are something which offer a unique way for the preservation of forms. So, wherever we have depositions, we have the chances for the preservation of the any form of life. Sometimes into the extruding volcanoes, some wood logs can be incorporated. So, in a very rare instance that we get fossils into the rocks other than the sedimentary rocks, but in majority of the cases we find them into the sediments. And the bigger the realm is, the lesser the disturbance is, the more is the chance of preservation of fossil. Then comes the way in which the fossils are preserved and the way they provide information about the biosphere existing at that time. That means the number of organisms or the biodiversity in a particular area is basically something which is not all preserved into the rock. That means, whatever information you get from the preservation of a particular layer is not the complete information of the biosphere existing at a particular time. That means, we get very partial information preserved. First, that all of the organisms are not preserved. Second, that the organisms which are preserved have only hard part of their body that gets preserved, like bones can be preserved, skeletons can be preserved, the shells, hard shells can be preserved, wood can be preserved, but the soft parts which also carries very vital information, they are not preserved. So, among the preserved animals also we have the partial preservation of their body, it is not that the complete body is preserved. Sometimes we see the complete bodies being preserved, we will talk about them a little later, but in the majority of the cases we see that only the partial preservation is accomplished. Then the third factor that comes into being is that even if an organism gets preserved into the rocks as fossils, also the rocks with fossils are denuded, they are destroyed, they are eroded, weathered, so they are removed. So, that also removes the information is lost to some extent. That means, we have a lot of constraints that control the information that we get about the biosphere with respect to the fossils as fossils about the biosphere existing at that time. The point is that in spite of the fact that we have a lot of factors that influence, we get some information which is very vital. So, even the preservation is a very rare phenomena. We will try to understand different components of this phenomena or the preservation. Sometimes the animals are preserved as such, nothing is lost from them, they are complete as a preserved form, complete form with all the soft and hard parts. The best example of such a complete body preservation is the woolly mammoth. If you see the woolly mammoth is something which was preserved accidentally, which was reported accidentally. The people in the frozen place of Siberia, they were searching some food to eat, but then they found that there is an elephant body within the ice and they started eating the flesh. Suddenly some of them they noticed that that this particular elephant bears a fur, and elephants usually they do not bear any wool on their surface. So, that was a kind of an interesting phenomena, interesting observation, that the elephants usually are without fur, but this elephant is with fur. So, he reported, I mean all of them, they reported that to the higher authorities, and then it was investigated and found that this animal is very old, thousands of years old animal got preserved as such into the ice. And it tells you that the earlier times the elephants they were having furs, and now the elephants have a different look. So, that is an example, that is the woolly mammoth in the frozen place of Siberia is one such very classical example of the entire body preservation, even the blood vessels were preserved. So, this is one way of preservation, entire body preservation. Another example of the entire body preservation that can be beautifully seen into the resins. Resins means the glue that comes, you would have very often seen that the bark of a tree or the stem of a tree has some liquids, they I mean blushing out of that particular surface. So, that forms a kind of a gum or the glue, and that is transparent. Some of the insects that get trapped into that glue, they get preserved as fossilized resins, and the mosquitoes and other insects have been preserved as such into the amber. Even the end Cretaceous rocks, they show the mosquitoes into the resins. So, and then some other places in the collieries we get the plant fossils and the resins which contains the insects that preserved into the resins. So, this is another example of the entire body preservation. Then comes another form of preservation which is called as the altered hard part. Hard parts are preserved, hard parts they are I mean having very less tendency of being degraded. So, hard part is preserved, but sometimes the hard parts are preserved altered, at other times the hard parts are preserved unaltered. I mean the minerals they change as the conditions change, like calcite forms from the aragonite. So, if the originally aragonitic composition shell gets preserved and becomes calcitic, then that is an example of the altered hard part. And when the calcite into the I mean shell of a mollusk, it remains calcite into the shell which is preserved as fossil into the rock. If both are calcite, that means the hard part is preserved unaltered. So, there are two categories, one category is without alteration and the next category is with alteration. The alteration can be mild, the alteration can be severe, sometimes the alteration cannot be even perceptible, but it is very commonly thought that if the preservation has to happen, the alteration is something which cannot be avoided. And it reminds me to share one of the ideas that we cannot call a recent shell as fossil. Like for example, if you go to a seashore and take a shell which is found among the sand, and you cannot regard that particular shell as fossils, why? Then it is preserved, it will not be degraded, but even then that is not a fossil. The I mean the shell of a mollusk or the shell of a lamellibranch or the gastropod which you pick from a river bed is not a fossil. However, the soft part has been taken away, only hard part is left before you. You can identify the feature, you can identify the animal from the morphological features of that particular form, but even then you cannot regard that recent shell as why? To be called as a fossil, a particular form has to undergo a process called as fossilization. That means from the biosphere it has to travel a journey to the lithosphere. So, from the biosphere it will be transported via some medium and it will be deposited into the layer will subside and the sand will be converted into a sandstone. the mud will be converted into a mud stone, lime will be converted into a limestone. So, by the process of the conversion of the sediment into the sedimentary rocks, that particular form will also be lithified along with the sediment into the sedimentary rocks. So, that diagenetic process, that process of lithification also works on to that fossil form. And if a particular form has this kind of a journey, undergoes this process, will only be called as a fossil, other forms will not be called as a fossil. That means, to be called as a fossil, a form has to undergo the process of fossilization. That means, when you see the fossils next time or a recent shell, then you will be you can take this into mind, what to call as a fossil and what not. Moving ahead with our discussion about the preservation of the fossil forms, or preservation of forms to be called as the fossil. We have discussed so far the entire body preservation as woolly mammoth, entire body preservation into the resins, and the altered hard part and unaltered hard part. The next and a very common mode of fossilization is the mold, cast, mold. That means, the shell often gets dissolved and it leaves the impression of the shell onto the sediment surface. Like, for example, if there is a wet soil and I put my palm onto that wet soil and I remove my palm. So, my palms are not there, but the impression of my palm would be there left onto the surface. You can see all those lines onto that palm. So, this is basically the mold. Then what is cast? So, it is very difficult to differentiate the mold and the cast. Cast is something which we have mimicked and we put something close that, and as you make gujiyas in Holi. So, you have a cast and you take the mold of you fill maidas and then you close that. You produce from one cast, you can produce several similar forms of a different composition. So, that is something which is a cast. But if you take that cast and put another material, then that cast makes a mold. That means, first you get a mold from a particular cast, and if you make the mold from this mold, then this particular mold will serve as the cast for the next mold. Except this, there is no visible difference between the mold and the cast. Cast can be used to make molds. So, cast can be the original shell, the cast can be the impression of the original shell. So, even if you produce an impression from the material, as a material serves as the cast for that mold, and if you produce the mold from another mold, so the earlier mold will serve as a cast for the next mold. As far as the type of molds are concerned, there are basically two different molds, as we could see. The first is the external mold, if it mimics the external surface, and if it mimics the internal surface, that is called as the internal mold. And I mean, sometimes if you imagine that a particular layer of a particular fossil lies over a layer and then lies under a layer, what happens actually? Happening is that the external form of that shell would have a mold onto one layer, and the internal form of shell will have a mold onto the other layer. And what if the shell dissolves at the interface? Then you will have the external mold on one particular layer and the internal mold onto the other particular layer. Like, for example, if the shell lies at the convex profile on a bed, then the top surface of the lower layer will have the internal mold of that shell. That means, the inner surface of the shell would be molded onto the external surface of the bed. Over that shell, if you have another layer, then the lower surface of the upper layer will mimic the upper surface of the shell. So, this is how the molds are formed. Sometimes the shell, many of the times say the shell dissolves, and then all we get is a mold. This is to be understood that if a particular mold mimics the external surface, it is called as external mold, as I told you, and if it mimics the internal surface, it is called as internal mold. Sometimes you have the deposition as nodules. Nodules form by the process of the diagenetic segregation. That means, in a particular layer of the carbonate or the lime, the chert is segregated as a particular bulb like form, which is called as nodule. So, it is a kind of a silica gel that segregates from a lime, and in that gel the forms are preserved beautifully. So, that is a nodule which is formed by the process of diagenetic segregation primarily. Therefore, the chert nodules in any layer, they are the having higher chances of the preservation of forms. So, whenever you go to the field, you find a layer of limestone and a chert nodules, always sample that nodule, and if you thin section that nodule, you will get beautiful fossils in many cases. I do not say that in every case you will be finding fossils, but there are more chances that these nodules would be fossiliferous. Then coming to the molds and their association with the nodules. Say for example, if a particular shell is covered by the nodule, then the fossil remains in between, and two sides of the nodule will mimic the two external surfaces of the shell. So, once you break the nodule into two compartments very carefully, so that the nodule does not breaks into smaller pieces, if you open up that nodule, then you will be finding the fossil in between and the two external molds of the fossils on two sides. So, you will be getting the hard part preserved at the same time, and the nodules will be preserved. Ammonoids are usually preserved by this fashion. Then after completing this particular preservation method, which is naturally existent, then comes the other method, and this other method is basically the method which is the best form of the fossilization, and that is called as the petrifaction. Petrifaction is something which preserves anatomy very beautifully. That means, if you consider a wood log, the wood log, if you take a transverse section of that wood, you will get the individual cells of the tissue. Sometimes the cells are hexagonal, sometimes the cells are pentagonal, the cells of the pith are different, the cells of the cortex are different, the cells of the epidermis are different. When this particular system, and then if you take a longitudinal section, you will be seeing the longitudinal cells of the tracheids and vesicles. But when you try to preserve such a system into the rock, what happens? The silica gradually replaces the tissues, the organic matter. The cellulose is replaced by the silica, but the structure remains same. That means, that kind of a wood after preservation would look like a wood, but the composition is siliceous. Its composition will be silica. It will, because it has been lithified, it has been converted into a rock, it will become heavier, the specific gravity will increase. But if you try to see the thin section, there will not be any difference between the real wood and the fossilized wood. So, that is the beauty of the petrifaction process, that does not alters the anatomy. No other method of fossilization offers such a unique opportunity to to study the anatomy even after the fossilization. In other cases, the anatomy is very difficult to be studied. So, this is one. The bones are they are fibrous structures. So, bones are lithified by the process of permineralization, that is very equivalent to the petrifaction. But in petrifaction, silica is the essential mineral that replaces the tissue. In permineralization, there can be other minerals that can do that. So, there is a minor difference between the permineralization and petrifaction. Then comes the carbonization. Carbonization means the volatiles would be lost and the thin film of carbon would remain on the surface. That means, it is not an impression, rather it is a compression. So, if you have the leaf on a black shale, so all the volatiles from the leaf would be removed by some sedimentary process, by higher temperature processes, and then you will have a thin film of carbon. All of us, the entire biodiversity on the earth, they are very different. Each animal is different from the other. Even in humans, one clan of human is different than the others. African are different from Chinese, Chinese are different from Americans, and we Asians are different from them. So, we have different communities even in the Homo sapiens. That means, there are diversities at several scales, but then if you make everything, plant, animal, everything as an organic matter, and if the organic matter decays to the full of its phenomena, what you would remain with is with the 5 elements: carbon, hydrogen, oxygen, nitrogen, sulfur and phosphorus. And if you arrange them through the valencies, it will be 1, 2, 3, 4, 5, 6. That means, the entire organic matter, it is preserved as a very simple elements with very simple atomic numbers, and this is very common with every organic matter. That means, the carbonization process, it leaves carbon and then other volatiles are removed. So, that process is called as carbonization. Leaves are preserved into the rocks by this process, carbonization. Leaves are preserved also by the impression, but by the compression also. Graptolites are usually preserved by the process of the carbonization. Graptolites are usually called as facies fossils also. Why facies fossils? Because they occur into the very fixed lithology. Other fossils can be found in a sandstone, in a limestone, in a shale, in many diverse lithologies, but facies fossils are the fossils which are confined only to one lithology, like graptolites will be found only in the black shale. In no other lithology you can find the graptolites. Yes, there can be 1 or 2 occurrences out of thousands and thousands of occurrences, so that can be taken as an exception. Otherwise, they are mostly confined to the black shales. So, this is what basically is called as the facies fossils. So, we had many different ways of fossilization. So, try to identify the fossils which you see into sedimentary rock in the field or in your laboratory. Try to find out what method of fossilization it has adopted. Different fossils are preserved by different methods, and it is our responsibility to segregate all these. If you have a new method, tell me. We all have existing methods of fossilization, but I do not agree that this is the only methods in which the nature preserves forms. There may be many methods which we may not be knowing at this stage, but this is the only domain, this is the only variation that we know which is commonly adopted for the preservation of forms into the rocks. I hope this lecture was clear to you. We discussed many different preservation themes. We discussed by the processes in which the fossils are produced or fossils are preserved, the forms are preserved as fossils into the rocks. We will be discussing the other paleontological groups in the lectures to come. Till then, thank you very much.